but it was the first successful DNA sequencing in space. It worked great.
Okay.
This ejection of mitochondria showing up in the bloodstream, and so it's a really fascinating feature of spaceflight, which we saw first in the NASA twin study. So, for in this case,
Mhm.
using a species that's been slightly modified to absorb, digest and basically process waste and upcycling, but we could see evidence of brain-associated peptides showing up in the blood, which is fascinating. The termite genome doesn't have the
Mhm.
ability to process the cellulose when it eats wood, right? So it's actually
Mhm.
coming from microbiome. Yeah, in ancient DNA, we worked with Oded Rachavi to look at the Dead Sea Scrolls, for example,
Mhm.
which we wanted to use the genetics to reconstruct some of the.
Welcome to the Omar Alayli Podcast, your go-to podcast for exploring the diverse fields driving space exploration and technology. I'm your host, Omar Alayli, and today I'm thrilled to be joined by Chris Mason, Professor of Physiology, Biophysics, and Genomics at Cornell. His pioneering work on how spaceflight alters the human genome, epigenome, and the human microbiome has reshaped our understanding of astronaut health. In his book, The Next 500 Years, Chris lays out a bold science based biological roadmap for how humanity can truly thrive as we expand across the solar system and beyond. Chris, it's great to have you on the podcast. Thank you so much for making the time.
Thanks, it's a pleasure to be here. Thanks for taking the time.
Chris, before we talk about spaceflight and its effects on the human body, I would like to start with something fundamental. Most of us walk through life thinking of ourselves as individuals and our bodies as a collection of organs like heart, lungs, brain, skin, etc. But in fact, as your work shows, it's a far more complex reality. Could you reframe our understanding of the human body not as a single entity or a collection of organs, but as a bustling ecosystem, a superorganism teeming with trillions of other life forms? And how does this change how we understand ourselves?
Yeah, happy to. So, and again, thanks for having me. It's a pleasure to be here. I think with what I termed in my book, the first book I wrote is like the meta species, or what some call the holobiont, or this concept that there are many species. Obviously, when we look out our window, or walk through a forest, or even see a beach, we can, of course, see organisms in the water. We see crabs on the beach. We can see birds in the sky. So I think most people already have this innate sense that we are in ecosystems, and that we have these complex interactions between all creatures that we can see and many that we can't see. But in our own body, it's the same basically construct, where we have, of course, human cells, which are defined by our chromosomes and genetic code, and there are tens of trillions of them. But there are also tens of trillions of microbial species on our skin, in our gut, and in our environment that we constantly interact with. And so I actually even tell medical students, if you study human genetics and you only look at human DNA, you are a bad human geneticist, because so much of our biology interacts with or is modified by, or even diseases can be caused by, viruses and bacteria, and of course, any interactions with the environment. So I try to always frame, the questions around what is the driving molecule or cell, regardless of what species it may come from.
Okay, so now let's take that concept of a finely balanced superorganism and place it into the most unnatural environment imaginable, space. From a high level, how does spaceflight begin to disrupt this intricate biological network you've just described?
It is a stressor that is unfamiliar, of course, to the biology, because we've grown up on Earth as a species. We've evolved to adapt to 1G, or gravity, here on Earth, and it really puts stress from a number of directions. There are a lot of fluid shifts, so you have liters of fluid moving through the body in unusual directions. There's, of course, radiation. There is the microgravity. There's the change in the environment that you have just in the walls of the space station, for example, or spacecraft, which is distinct from what you see here on Earth. And it just creates a lot of, almost molecular confusion, or at least a bit of a priming of alertness that the body has to adapt to. And so what's extraordinary is that there is consistently a series of cytokines, which are these signaling molecules between cells that can indicate inflammation or stress. They are when you take off and when you land to go to space. But almost all people, once they're in space long enough, sometimes it's a day or two, sometimes it's a week, but the body does adapt, actually. And same thing when you get back to Earth. There's a day or two of really significant readaptation, but then the body is quite resilient. And so that's one thing we've taken a lot of comfort with, is that even though we see thousands of genes changing expression, or these cytokines, or molecular changes and microbes changing, many things changing, but the vast majority of them do reset and seem to reacclimate, which is a good sign for some of these longer missions.
Okay. To really grasp the complexity of what happens during spaceflight and understand the fascinating yet unexpected results from your spaceflight studies and research, I would first like to set the stage for our audience by exploring a hypothetical example of a model that links several complex biological systems as a response to an external catastrophic stimulus. This simplified model would be our baseline and represents a cascade of causational events that happens in the body as a result of and an attempt to remedy the effects of an external catastrophic stimulus, as I just said. So let's start at the very beginning of this cascade. Our bodies, let's say our skin, get exposed to radiation. It could be long exposure to the sun, let's say. Now, that radiation causes damage, and it causes damage and radiation damage, which leads to DNA and oxidative damage in the skin cells. At this initial point of impact, what is happening at the molecular level that kicks off this entire emergency response?
Well, when you say, yeah, it's a good example you gave. So just, in the sun, when you have the radiation, there's energetic particles coming literally from the sky that can hit your DNA and make what's called, often cases, a pyrimidine dimer. So actually can even fuse together pieces of DNA that aren't normally together. In this case, cytosines, pairing C to T's pairing together, like versus ACGT is the genetic code. But suddenly you have these pyrimidine dimers where C's and T's start to bind together and create these mutations. And also, you can have double strand breaks. You can have breaks of the DNA. You can have mergers of the bases of DNA. You also just have some of the radiation itself is energetic, so that could impact like mitochondria. So the influx of literally high energy particles, or at least in this case low energy particles, but enough that can actually cause some DNA damage or DNA stress, is a response to the body. Even when you get a sunburn, that is partly a response to the degree of damage and stress that's occurred to your DNA and to the skin cells. Now, we of course have an atmosphere that protects us. We have an outer layer of skin that absorbs some of that damage. We have many pathways that activate DNA damage repair pathways, or DDR pathways, that really set to work quickly within a day or two, fixing most of these problems. And we all get mutations every day. So this is something that happens all the time, every day. It's a part of being in the universe. But what's extraordinary about life and human life, as well as many others, is that we have all these innate pathways to see the damage, detect it, correct it, either with non-homologous end joining or end repair. There's different methods to repair
Mhm.
DNA, and they all just go to work. You don't have to, of course, think about it; it just happens naturally. And it's really extraordinary to see that this happens on Earth and in space. And so we see some of those same pathways occur in spaceflight as we see, for example, just from low dose radiation. That's just from like
Mhm.
a sunburn.
Okay, so now we have radiation damage leading to DNA and oxidative damage. That cellular damage then triggers the next step in our model, which is the altered gene expression, leading to the release of cytokines. This is a critical junction. The cell's software is essentially being rewritten in response to the damage, like you said. So this is a two-part question. Could you first explain the concept of micro RNA methylation? Like how are the instructions being modified? And second, what exactly are these cytokines that the cells begins to release, and how do they differ from hormones?
Yes. So there's both features occur in RNA. So small RNAs, what are called microRNAs, are these small, usually between 22 and 25 bases. So very small RNAs. Other bases
Mhm.
are thousands of bases long. So they're small regulatory RNAs that can help control gene expression. So some of those are changing when you get DNA damage. Also, RNA methylation. We see that's also small chemical groups that change. Essentially, the best analogy would be when you think about turning on and off a machine in your kitchen, for example, a blender. You turn it on, turn it off. But of course, the blender has different speeds, and so it's not just a question of on or off. It is on, but how fast? And so I think a lot of these mechanisms for RNA control can attenuate or modify the speed or type of expression you see, not just yes or no, but which degree. So we see these change, and then also we can see that these modifications then do lead to changes in gene expression for genes related to DNA repair or cytokines. You mentioned cytokines and hormones. And so to the second question, cytokines are the cell signaling molecules, which often are In many cases, they're pro-inflammatory or anti-inflammatory. They're trying to keep a balance in your body. For example, if you get an injury, you get a cut, you start to see inflammation by the cut, or you get a bruise, you'll see the bruising and inflammation. You bump your head, you'll get a bump on your head. So all these are features of inflammation being regulated. And the same thing is happening with these cytokines, whereas hormones are often what go into the nucleus of cells and themselves go and change the expression of other cells. So like for example, when you go through puberty, you go from having no facial hair to suddenly having facial hair or something. So it's going in and functionally changing other cells and often even going into the nucleus and really modifying them. Whereas cytokines are much more transient. They're just kind of a quick short-term response to changes.
Okay, so these cytokines are the first responders signal, I would assume, as
Yeah,
an
it's a good,
analogy.
they're the first ones that jump up and say, we're here to
Yeah.
help it.
So the next step in our model is the local inflammation and white blood cell recruitment. You described the cytokines as working locally. What is happening in that immediate localized area as these signals draw in the body's security forces, the white blood cells?
Yeah, these signaling molecules, of course, perfuse into the bloodstream and can signal to the bone marrow to change activity for immune cells. They can signal existing immune cells to be deployed to that area. If it's a damage, like where it's a wound, you can actually also get platelets to come to help, seal the wound so you don't bleed out, of course. So, all these factors are helping to recruit other cells to the site. In the case of the white blood cells or natural killer cells, these are cells that can help actually clean up the mess if there's an external agent. Now, in some cases, if it's just damage, they might come and see no foreign entity and then move on. But in other cases, they will stay and they will attack and basically, clean up the mess, I'd say.
Okay, so let's recap so that our audience are with us. So we start with radiation, causes radiation damage, then DNA oxidative damage in the cells, which leads to altered gene expression. Then skin cells release cytokines, which affects the local inflammation and start doing white blood cell recruitment. Now, if the exposure is continuous or severe, or the damage is severe, the situation can then escalate, right? This leads us to the inflammatory response being amplified by DAMPs, the damage associated molecular patterns, the mitochondrial DNA. This suggests the initial alarm isn't enough and the body needs to shout louder. That's why the body needs to amplify the signal. And what is the significance of something like the mitochondrial DNA being found outside the mitochondria acting as a system-wide alert?
Yeah, it's a good question. Yeah, and there are multiple pathways there. So there's actually, we've seen, you mentioned the DAMPs, the damage associated molecular patterns. There's also something called PAMPs, or the
Okay.
pathogen associated molecular patterns, which are also a way to signal to the body that there's damage or there's a pathogen or there's some crisis that's in a local environment. And the other interesting thing about mitochondria is when you release mitochondria into the bloodstream, they have what's called an interferogenic response, meaning they prime interferons, which are also signaling molecules that you see during infection, similar to cytokines, but very generally much more related to infections. And it's extraordinary because they help prime this larger response. And so we've seen it as a general stress response, but we've also seen it consistently in spaceflight.
Okay.
This ejection of mitochondria showing up in the bloodstream, and so it's a really fascinating feature of spaceflight, which we saw first in the NASA twin study. We saw it again from some of the Inspiration 4 data and some Japanese space agency JAXA data sets. So we keep seeing this, and it is in for a lot of these changes, it's very important. They sound like it's damage or infection or all these harsh terms, but the ongoing debate in the field is, are these adaptive or maladaptive, and we see many changes, but the ongoing debate and really uncertainty is that they are changes, but we don't know, like, if you were to prevent them, would that be a good thing or a bad thing? And I'd say we don't know enough yet to say, let's take an inhibitor for DAMPs, for example, or IL-6. I think it's too early to say whether that'd be good or bad, but we at least can now see the changes that happen when you go in space. But it's there we yes, whether they're damage or just response, like when you have a wound, you repair it, and so that process is a good thing. You want to
Mhm.
repair it. So we're trying to figure out how much of these pathways are good versus bad, or just even associated. Yeah.
Okay, great. And this brings us to the final step in our model. The brain senses the systematic stress, activating the HPA axis and causing the release of hormones like cortisol. the body's central command is now fully engaged. What is happening in this stage as the brain takes control, and what is the purpose of flooding the system with stress hormones?
Stress hormones, even though they do create a lot of increased heart rate, dilating blood vessels, you can have increased focus. The stress hormones themselves, though, are a way to prime the body to move, to take action. So the brain being involved is a good thing. It's always good when the brain's involved. I guess unless there's mental disease, then that's not as good. But broadly, having the ability of the body to be fully activated and ready to move, say to get medication, to get to a different environment, if it's radiation, to move to an area where there's not radiation or less radiation. So I think these are all then it becomes part of the feedback loop where the body is regulating its own regulation, and again, pharmacological interventions, or even just tracking and say, okay, I see many of these changes, and these levels of stress are within normal limits. So, for example, when most people go to the doctor, you have ranges for most of your molecules in your body, like insulin, for example, has a certain range, or different hemoglobin or iron levels. Like we want them. It's not you have to just be within a range. And so, if you're within the range, then you could say, okay, I'm probably okay for now. But then, if something's too high, then you could take action.
Okay, so now I would like to turn to the landmark NASA twin study that you participated in as a lead investigator. When we look at the data from Scott Kelly's year in space, this neat sequential cascade seems to be thrown a little bit into this array. Your work reveals several paradoxical spikes in the inflammatory markers. Let's start with the potent chemokine, the CXCL5. On Earth, we'd expect them to peak during the local inflammation. Yet in Scott Kelly, its levels were elevated almost continuously throughout his time in space. What does this persistent high level alarm tells us about the state of the astronaut's body in orbit?
Yeah, it's so a lot of these cytokines, we do expect them to stabilize. So actually, yeah, CXCL5. So we looked at several hundred different cytokines, and that was one that actually did spike up and stay higher than we would have expected. Like a lot of times, it does just revert back down to normal. And so I think it was surprising in the sense that we could actually see it spike up very early in the mission, but it was actually later in the mission. And it's a very nice nugget of the paper. There's a big paper on it, but one of them was that we looked for some things that spiked up and went down, others that seemed to really go down or up and like seemed to be very transient. But CXCL5 was actually one of the few cytokines that went up as sort of a stress response, but then actually it went up even more later in the mission, like after 6, 7, 8 months. So it's a very intriguing cytokine. And so, and like I was saying before, I'm not yet in a position with only N of 1 to say whether it's good or bad. Maybe this is the essential adaptive cytokine to help you survive in space for a long time, or it
Okay.
has no bearing whatsoever. We don't know enough from one person, but it's certainly, these early measurements and these early missions where we can monitor people at such great depth give us clues as to at least what are the responsive molecules, whether they're, again, adaptive, maladaptive, associated, or just coming along for the ride. We're still teasing that out, but it was really an extraordinary. And then when you got back down to Earth, it came right back down,
Mhm.
stayed down, and so it really showed that this seems to be pretty specific to spaceflight and pretty specific to the duration of spaceflight, which is very fascinating, definitely.
Does it have an influence for, let's say, microgravity versus radiation, or this is still an unknown?
Well, because the radiations, but so far there doesn't seem to be an association directly because the radiation was fairly constant during the mission. It was a total of 147 millisieverts of radiation of a dose over about the year. And so we don't know yet. And that's something that these are the questions where as people go into missions that go farther into space, we can start to tease out, okay, there was this amount of radiation and these molecular changes and start to associate them. We've been doing some of this with simulated experiments where we have PBMCs from blood donors and then irradiate these cells and look at genetic differences and other responses. So we're beginning some of that work in vitro studies on Earth with simulated radiation, but eventually getting as much other data from other missions is what we want to do for sure.
Cool, very interesting. So then there's the cytokine, the interleukin 1 receptor antagonist, the ILRA1. Again, we'd expect this cytokine to peak early on after the cells change gene expression. But in Scott Kelly, the major spike occurred after he returned to Earth. What is your leading theory for why this crucial inflammatory response was delayed until his return?
Good question. So I think, yeah, and that was one of the cases where, from some of our published work, That was it was the highest level we'd ever seen from someone who'd returned back to Earth. And to date, that's still true as far as our records have. It's a great question, and it was such a striking difference. So we also double checked the data, double checked the technical replicates to make sure the data was real, and so he we're not sure, except that it was the longest mission that, of course, any American astronaut had done up to that point. It was maybe related to the fact that it is a long mission, that could be a factor. We of course don't know for sure. We did see some of it also change a little bit from short duration missions the days after. So this may even be one of our papers called, like, what are the returning to gravity markers? Like, okay, your body's coming back to gravity. You need to basically rebuild muscle, prepare the bones for being back on gravity. So this, in terms of a candidate molecule, something that helps you get back to gravity, this would be the one that we'd probably point to, definitely, and talked about in some of the papers.
Yeah, and another one that is also interesting is the C-reactive protein, the CRP, which is also which also spiked also post-flight. But what does this delayed 1-2 punch of the ILR1/6 then CRP tells us about the profound physiological shock of readapting to Earth's gravity?
I think it shows that, and CRP is something we also measure for, like, aging markers. It's linked to other damage, like there are people that have it for inflammation, infection, accidents. So I think it is. It may not be the only molecule that's helpful, but it is something that has the part of the body that says, "Oh, we're back to gravity. We have to really adapt." What's interesting is as you just mentioned, we didn't see it really at all when he first got into orbit. This seems to be something that was quiet until he landed. And so another thing here is it didn't just spike up at one time point. It was the day of landing plus a few days later, and then came back down again. So we could see some of these changes that I just also get more confidence if I see something at more than one time point. Of course, if it just is a blip, there could have been with double technical errors and make sure things are running plates. But some of the more interesting patterns are when we see these multiple time points. So we have a lot of opportunistic collections we've done over the years, but we want to start to get samples collected, daily, potentially, for some of these missions, to get a lot more high resolution changes, because that would help us answer some of these questions for sure.
Yeah. So speaking of a blip, perhaps the most perplexing spike was the thyroid stimulating hormone, TSH, which occurred just before Scott Kelly's return to Earth. This is typically part of the endocrine stress response, which our model we discussed happens at the end. But a thyroid response seems very specific and out of place. Why the thyroid, and why at that precise moment, just as he was preparing to come home? And what does this suggest about the body's anticipatory stress response?
Yeah, it's a good question. So there is a big mystery. We're not sure. That was something that we haven't seen in any other missions, and so maybe leading theories, it's something very specific towards these long duration missions. So that's one of the I think like a lot of science, we get some answers, but then we get a lot more questions, and that's one that's still an open question, I would say, of what is happening, and since it's so unusual, the guess is that it's probably related to the fact that it was such a long mission, but that's of course to be determined a bit.
Okay, because TSH also is responsible for metabolism. Is there something he was eating? Maybe also changes in his diet at the land. Maybe I don't know.
That's a good question. So we did ask at one point. We said, could we maybe get Mark and Scott Kelly to both eat the exact same food to help try and normalize some of these differences? We did not quite do that. That was requested, but was declined as part of the protocol. But it was, because it'd be not just for a week or a month; it'd be for like a full year of eating dehydrated food on Earth, which is not appealing, of course, if you're on Earth. So it could be food. The medication, there weren't too many medication differences, but there were some differences there as well. Some of them just for sleep medication, which almost every astronaut takes. But there was no obvious difference, I guess, in really in any of these differences. So I think and Scott Kelly wrote about some of that in his book. Other factors he talked about as using medication to get some sleep, but he also talked about carbon dioxide spikes. In some cases there was evidence of hypercapnia, where there's too much carbon dioxide because the CO2 scrubbers had to be fixed. So there's many factors that could be contributing that, of course, are different. So we're still looking at all of them together.
Interesting. So the twin study was foundational, but you've since analyzed data, like you were just telling us before, from a much broader range of human experiences, from trauma experiencers to high altitude climbers to the first all civilian inspiration 4 crew. First, are you seeing that same pattern of genetic and epigenetic disruption across these different forms of extreme environmental stress? And second, what does this tell us about how our bodies are fundamentally programmed to respond to any environment that isn't Earth normal, knowing that Earth normal might as well be from zero sea level to like 2 kilometers, 1.2 miles above sea level?
Yes, yeah, like that. Well, so yeah, we did see some of the consistent trends. So, for example, IL-6, we saw a change again. We saw elongated telomeres. We saw a lot of the differences in some of the same cytokines. We saw also gene expression differences, so including DNA damage repair and just genome maintenance. So
Mhm.
we do see some consistent features, but we also saw that there was some evidence of a dose response. Some of the changes were present, but at a much lower level than what we saw in the twin study. So I think the time that you spend in space may also be a factor as to how much you actually will have some of these molecular phenotypes appear. But it is exciting because we're starting to see these civilian missions. We're starting to see an opening of more spatial. The Polaris Dawn mission just finished. That was an orbital mission. There's plans for a lot of new space stations in the next 2 or 3 years. So I think we'll see a lot more opportunities like the ones we just saw for the Inspiration 4 mission.
Okay. What about about how body are fundamentally programmed to respond to any environment that isn't Earth normal? Is there any revelations here? That is because I've heard that also the separation for a crew went a higher altitude, so they also got maybe similar data to people who have been longer in space, just because they went a bit higher. Is there a connection between this?
It's a good theory so far, but we don't quite know yet. I think it seems like possibly there's definitely
Hmm.
like a correlation. So we see some differences, but I can't. There's just we just don't have enough data to say that it's really. I would even say there's not even enough data yet to say it's a trend. I'd say it's an observation that's interesting and makes you want to watch it again for future missions. But we saw a lot of differences between different crew members, like for CCL2 or IL-10. Again, we see in many cases most of them go up, but occasionally some of these markers go down, or we think it's both the crew member and the mission duration that are pretty big factors that we're just teasing out. I just had a meeting last week about this, and the discussion was how many astronauts do we need to monitor before we can start to have really good predictive metrics, and the answer is about 10,000 before we could really have statistical power to start to make strong recommendations. But we see a lot of interesting observations right now, and that's what we're watching for these future missions for sure.
Okay. Let's now shift to the epigenetic and brain development and relation it to spaceflight. We've discussed systematic inflammation, and I want to connect that to one of the most critical internal systems that we have, the human brain. There's a well established link between inflammation in the body and the brain health, often called the gut brain axis. Can you just give us a quick overview of what is the brain or gut brain axis, and how does the systematic stress and microbial disruption you see in astronauts potentially translate to challenges for brain health and the cognitive functioning during long missions?
Yeah, there's a lot of connections. I was taking at the very beginning of the podcast, the trillions of organisms that contribute towards our physiology that we know are big factors, and I think we've been looking at this in mouse models and human models. This is something that we know impacts how you regulate glucose, how you process drugs when you take a pharmaceutical. It's either modified by or sometimes completely made anew by and processed by a microbe. So the gut brain axis is a key feature, now recognized feature of human health. And we've seen actually a lot of the microbial diversity in the crews that we've looked at has been mostly maintained, but we do see ratios of organisms change in flight. For example, the ratios of different firmicutes types of bacteria often will increase, and which sometimes is associated with poor gut health, but is again here it's too early to say is it really driving it, but we do see changes, and I think this is something we want to keep in mind and monitor for every future mission.
Okay. You've established in your research that some short duration flights show molecular signs of potential disruptions to the blood brain barrier. How concerned are you about this, and what are the implications for an astronaut's risk for developing long term neurological issues?
Good. I'm not concerned yet because right now it's a very fascinating observation. But most crew members are very healthy and have been monitored afterward, and they are generally cognitively okay. There's no obvious sign of long-term neurodegeneration or neurodevelopmental or neurodegenerative disorders that have been appearing. So Keeping in mind we only have a few hundred astronauts, so we don't really know for sure if there's a strong association. But we could see evidence of brain-associated peptides showing up in the blood, which is fascinating. And then when we look in mouse models for mice that had flown in space, brought them back down, and this was with Vivian Mao, dissected the brains, looked for markers of blood-brain barrier integrity. We could see that those markers changed for the space-flown mice, and we could see some disruption evidence
Okay.
of the mouse blood-brain barrier, which again is not the same as the human. It's a little bit imperfect, but we can at least see there's some differences to be tracked. And so I think what's exciting is you can monitor in the blood through exosomes what's likely coming either from the brain or proteins related to the brain, and it serves as a way to do somewhat neurological monitoring in terms of sort of the structure, not so much like what they're thinking, but you can actually get a sense of the integrity and structure. So that is very new data, and again, I'm not
Mhm.
concerned yet because right now it's just a handful of observations, but it's interesting that we can see some of these differences and now monitor them, track them, and then use them. Yeah.
Okay, you've described the brain as having a molecular recipe. If spaceflight is constantly altering the ingredients of that recipe through epigenetic changes and inflammation, what happens to the gut brain axis when microbes and neurochemicals flow differently in microgravity?
There is this recipe that changes and adapts in spaceflight. The microbes themselves also undergo selection pressure and also have changes in their structure. So we do know that there are changes that can and do occur, and it is. But it's the microbes are changing some of their expression levels as well. We even looked at yeast that have flown in space. I've looked at microbes on the skin, bacteria, for example. Usually it's studied in different domains. Normally, if someone's doing a human spaceflight study, they've historically just looked at the human data. If someone's looking at microbes, they just look at microbes. But only recently, like with the twin study, we started really combine some of these metrics, and we did this for the I-4 mission as well. So that's where we're beginning to understand it, but I think there are a lot of changes, but I don't know which ones are the most important yet for the humans. But the microbes are changing just as much as the people are, if not more in some cases.
Okay, so what early indicators should mission planners and medical officers monitor to prevent cognitive decline on long voyages or long term missions?
I think some of them would be these markers for, if you start to see really large changes in blood-brain barrier markers, that could be an indicator. Cytokine levels would be good to monitor. You could imagine if you look at cell-free nucleic acids, like cell-free DNA and RNA, we've published a lot on. You can see which tissues are undergoing lysis or damage. So that's another way where if you suddenly see a spike of an unexpected proportion of a tissue showing damage, that could be of interest. And of course, there's other things like the complete blood counts or blood chemistry, do some basic metabolomics. You can look at nutrition levels. So other factors you would definitely want to monitor that are sort of standard for blood pressure, heart rate, always more basic measures. But they're combining them all together gives you a real sense of how perturbed a particular astronaut might be during a long-term mission.
Okay. A recurring theme in your work is the shift from reactive to proactive medicine. You took a portable DNA sequencer, the MinION, and got it working on the International Space Station. This fundamentally changes the game from analyzing data months, maybe sometimes later on Earth, to potentially diagnosing the illness in real time in space. What is the biggest non-obvious challenges in making that technology work in zero gravity, and how different is Minion as a DNA sequencer from anything on Earth?
Yeah, so what's very exciting is this is with Kate Rubins, who was an astronaut that was in flight then, with Dr. Rubins, also with Sarah Wallace from Johnson Space Center, and Aaron Burton. So it was a whole team effort. Charles Chu was involved as well. So we had a whole team get together, say how quickly we've got a chance to fly this in orbit and get it actually up there and working. So the biggest difference is it's very small and light, which is important for spaceflight and for remote diagnostics. So it's the size of smaller than your cell phone if you have a phone. It's even smaller than that, and so it gives you a chance to take a very portable, highly integrated technological device and get it in flight. It also sequences. It's a nanopore sequencer, meaning it moves individual molecules through a small pore,
Hmm.
the size of in the nanometers, nanopore, and it does pull that individual molecule through, and then you can monitor what bases are present as it moves through the pore, as it transits the pore.
Hmm.
And it gives you a sense to look at the raw molecule. You can look at epigenetic differences. You can get very long reads. So it's different in a lot of those really good ways from other kinds of sequencers that are on Earth. And yeah, we flew it. We did a parabolic flight test in 2015 that succeeded, and then flew it in orbit, and then published that work, about 18 months later. But it was the first successful DNA sequencing in space. It worked great. To your other question, the biggest challenge was getting automation for extraction and preparing that sample for sequencing. The first time we did it, we flew libraries that had already been made to show that it worked. It worked fine. Since in other experiments you can do the library prep in orbit, other
Hmm.
sequencing methods. So that we've now done other variations of this, either in remote areas on Earth or in parabolic flights or on space stations. And there's an active plan to put these into all the upcoming space stations as well. So you have this real time sequencing capability that's standard of operations for most space stations.
Super cool. Uh, Now that we can sequence DNA and RNA in space and tracking the biomedical roadmap you drew in your The Next 500 Years book, can you define which milestone we are currently at compared to that to your book timeline, and what is our next milestone according to your roadmap, and what is the next logical technological leap we need to achieve it?
Yes, I think we are in what would currently be called phase two of the plan, which means that we had done preliminary work of understanding the human genome for
Hmm.
10 years up until 2020. And then the goal for the next 20 years, including now we're about 5 years in, was to improve all the methods for rapid sequencing, but then also genome engineering, and which at the time when I wrote the book, this point, 5 years ago, it thought that it would be harder to get CRISPR and other genome modification systems to be as well deployed, but now they're being used in clinical trials, and genome engineering has been deployed for several diseases successfully, and one that has an FDA approval. So it is, when I first sketched this out in 2010 as a bullet point list on my lab website or an IGM website,
Hmm.
it was very abstract, and I would have thought it would take much longer to get large scale genome modification and manipulation completed. But now we've seen treatments for sickle cell disease, beta thalassemia, different inborn errors of metabolism at birth,
Hmm.
CRISPR therapy that was just deployed a few months ago. We've seen the dire wolf, multiplex genome editing for de-extinction efforts. That's something that was done also a few months ago, which we contributed to. So it is it's a fascinating time, and so I think, in some senses, we're further ahead than I thought we'd be, actually.
Hmm.
But in other cases, the ability to be on the moon or on Mars is I think a bit behind from where we thought we'd be in 2010, for example. We thought we'd be back to the moon by now, and maybe even have a moon base, but we're still getting there. So it's both faster and slower than some expectations of what we had.
Okay, so we have the DNA sequencing, and we have CRISPR almost ready. What's the next milestone do you think we should achieve?
Yeah, there I'd want to see more basically contained systems for surviving on the moon, or someday Mars, but what's often called in situ resource utilization, or ISRU, are
Hmm.
these self-contained systems where you can take all of the human waste and recycle all of it. Actually, the ISS does a great job of collecting most of the water. It's about 97-98% successful for reclaiming sweat, humidity, moisture, urine. It all gets recycled. But a lot of the carbon dioxide gets vented into space, or some of the nitrogen that's in urea and urine. We don't use that for fertilizer. So there's plenty of examples where we could really have a nearly closed loop system that would maintain all the infrastructure and atomic utility, if you will, that's present for these longer missions. That's where I think a lot of the challenges still remain.
Okay, so you're saying in a way that in integrating the current science and technologies into other disciplines, so that to make it a holistic closed loop approach. Okay, okay. So Chris, we've touched on the human as a superorganism, and I would like to dive deeper into the super part, the microbiome, the microbial ecosystem within us all. Um, to give our audience a sense of scale, can you frame? I know you touched on that in early, but here, can you frame just how vast this microbial world is within us, and beyond just existing, what are some of the most critical jobs that these microbes are doing for us every single day?
Yes, there are. So the numbers have been debated for a while. At one point, it was thought that the human body has about 30 to 40 trillion cells, and that the microbes are maybe 50 trillion or maybe 100 trillion. So over the past 10, 15 years, it's been debated a few times, and there's various
Hmm.
estimates. And also that there's more microbial species, of course, in their gut, because there's one human species on your body, but then there's thousands and thousands of microbial species. They each make different gene products, so They make more genes than we do, or at least types of genes. So that's also fascinating. There's also bacteria and viruses and sometimes fungi, for example, on skin. So there are multiple domains of life all interacting, working, making more kinds of biology than we could make in our own genome. But more recently, the estimates are it's more about maybe about 50 to 60 trillion human cells, probably about 50 to 60 bacterial cells. So it might be about even in terms of
Hmm.
numbers. But the numbers, don't matter. You can have, one single virus like an Ebola that you don't care how many there are. They're catastrophic, right? So you don't need to have, it's not a numbers game of how many cells you have and who has the majority, because even a very tiny minority can wreak havoc or completely, of course, change your biology. But we've seen, a lot of them are, Bifidobacterium or Akkermansia are two different genera of bacteria that have a really extraordinarily powerful feature. For example, in our gut, they are keystone species that can regulate glucose levels and also can stabilize gut diversity. So there are, some of these key species that have such a powerful force. You'd want to make sure you maintain them and keep them healthy in your body. And I think that's something that we are going to, continue looking for in terms of how much we see these species that if you remove that species leads to a lot of problems. Now, they also make individual molecules. They make a lot of, basically human mimic type molecules where the body can respond to and can also sometimes lead to inflammation. I look more at the species level because each species, some of them are really powerful species all on their own, I'd say.
Yeah. I read somewhere that actually cows, while they eat grass and it goes to one of the stomachs, the grass feeds the bacteria in its belly, and then it ends up throwing those excess bacteria to use those bacteria as proteins for the body. So yeah, this is really important. The microbial ecosystem.
Yeah, and the cellular, like, another great example is termites. For example, the termite genome doesn't have the ability to process the cellulose when it eats wood, right? So it's actually
Hmm.
coming from microbiome. And when baby termites are born, they have to get a microbial transfer from the mother, quite
Mm.
literally from the butt of the mother, to pick up this like probiotic that will help them digest wood. So it is, I think it's a little bit gross, but it is how they survive, right? So
Hmm.
it is a microbial driven phenotype that they get from their mother when they're babies.
So, if these microbial communities are so vital, how does spaceflight, with its sterile environment, specialized diet for the astronauts, and radiation, lead to dysbiosis, or the imbalance in the ecosystem?
Yeah, in a lot of ways we don't understand. So the stress in the body, the cytokines that humans make can and do interact with the inflammation, like in the gut, for example, which could eject different species or modify their environment, so that'd be less likely to stay potentially. So there is a continual back and forth between the microbes in the gut and the intestinal lining and the skin and the inflammation there. So we don't have a lot of data yet and say, we see changes, but is it just because the diet was different? There's too many cofactors and variables to know yet. Was it this one change that definitely led to an ejection of a species or a change of a microbial peptide? Or do we see, sometimes we've had studies that show more antibiotic resistance for bacteria and some that have shown less in spaceflight. So we've seen both as well. And so it's like most things in biology. The answer will almost always be, well, it depends. It depends on the context, the medications, the environment, humidity, temperature, all these other factors. And so, at least for microbial specific response, we do see adaptation. We do see that they're evolving, as all life does everywhere it goes. But there's no one. It's not as consistent of a feature profile yet that we've done. The biggest data set that's ever been made for the metabolites of microbes was just published a couple months ago. So we're still looking at that data now and comparing it to some of our old spaceflight data. So a lot of it we just haven't had the data to even know yet, but we're looking at it.
Okay, so for our audience who are not like very much into, haven't studied medicine, what are the tangible, like an example of health consequences for an astronaut when their internal microbial partners are thrown into this array? Is there any anecdote you can share?
Yeah, well, we've done. I think we take the best lessons for space from the ones we got from Earth. So, for example, different probiotics that have these keystone species, like Bifidobacteria or Akkermansia, or have some of these, well-known, these generally considered positive species, can help replace the gut ecosystem. We did a clinical trial where we looked at people's gut species, sequenced this tool, looked what was missing, and then replaced it, and saw that decreased inflammation scores. And so I think we've not yet deployed that kind of a protocol for spaceflight, but we've done this on Earth. We know it has a tangible benefit. The patients improved. we have seen other clinical trials that have validated this result, is that if you can see what's missing, you could engineer and or give the missing microbes to or especially when you compare to a baseline. We can say,
Hmm.
okay, you are stable, you look healthy, and you go to space, suddenly you've lost all these species that were with you for years. Let's put them back. And so that, conceptually it's ready, but we just haven't deployed it yet in space. But that's something I'd love to see in the coming few years, which actually wouldn't be too difficult, because you'd pick the selected species that are likely to be beneficial. Now, it's basically a clinical trial. You'd have to register
Hmm.
this and run it as an actual trial. But I think we know enough now to at least to begin to plan that out, and I think start to try it.
Cool. So, your work on the Inspiration 4 mission revealed a fascinating phenomenon you called microbial blending. It seems that within days, the crew began to share microbes, creating a unique spacecraft microbiome. Um, what are the implications of this?
Yeah, so some of it was just was intriguing, just to see that they became more similar to each other in the microbial space, which we observed for other studies. We've seen this for Jessica Green has studied, for example, people who play roller derby or people who play basketball. When you bump
Hmm.
into people and you have a very confined space, you will have a microbiome transfer. And
Hmm.
so some of it was an expected hypothesis that we would see some of this, which we did. But the other thing that was surprising is if you track by where we see the number of species and where they're moving from which part of the body, because we sampled 10 parts of the body, and we built out this protocol for a 10 sample collection, which has now been used by several other groups. We get a really good map of what's moving around the body, within someone's body or also between
Hmm.
other crew members. And
Okay.
the armpit was the place where we saw the most movement, if you will. So that was also kind of fascinating for that mission. So we've deployed this now for several other missions, and are in the middle of sequencing and getting some of the data back on that. But we'll have a better window into that probably the next year or two, because it's fascinating to see. And we also saw it for the twin study. We could actually see that the microbes in the space station wall seem to be interacting with the T cells from Scott Kelly, for example. So what's fascinating is we can start to look at the immune system and the microbes and how they interact with each other.
Okay. So, does that mean if one crew member gets sick, the entire crew is more vulnerable, or could it potentially lead to a more robust shared immunity?
It could be. So that's we don't know yet, but it's likely. So, for example, if someone had a cold, like a rhinovirus, and they're
Hmm.
in that small of an environment, it would be very hard for people not to pick it up. Now, they could pick it up though and have no symptoms, right? They could get the virus and just be fine, as happens all the time on Earth. So, just because you get a virus doesn't mean you'll get sick.
Hmm.
Just because you're exposed doesn't mean you'll get the virus. There's many layers, of course, to go through before you get sick and get the virus. But you certainly would be at very high risk if you're in a sealed spacecraft and someone has flu or COVID or a new
Hmm.
virus. It would be it'd be a high risk environment, I'd say.
So, beyond the astronauts themselves, you've also mapped the microbial landscape of the International Space Station. You found that some bacteria, like the Acinetobacter piti, am I pronouncing it right?
Mhm,
Have evolved in space to become more resilient to antibiotics. This sounds like a beginning of a science fiction horror movie.
mhm.
How do we manage the evolution of these superbugs in a closed environment, millions of miles from a hospital?
Like the Andromeda Strain by Michael Crichton, or some other recent books or movies. Well,
Mhm.
no, so we see it was evolving, and it did create a different clade relative to other Acinetobacter ptii that we've looked at. So it was what's interesting though is some of the genes that changed were not associated with virulence, but they seem to or with resistance to antibiotics,
Mhm.
but they seem to give this phenotype. So things like when we look at a gene and we think we know what it does, oftentimes it's based on the limited data you have. And so, of course, sometimes one gene can have multiple functions, what the term is called pleiotropy in genetics, that one molecule can do several things, much like a Swiss Army knife can do multiple things. It's not just a knife, it's also a screwdriver, it's also a prying opener, it's a bottle opener. So certain proteins are like that and genes are like that, where they do many things. And so we've begun to see that the genes that weren't originally annotated or described as something that leads to resistance can do so. So that was one interesting feature. But then also that it is evolved. It's a different, effectively a different species at this point. So now we don't think everyone's at risk on the space station, but it does mean that there are significant differences there that we should track, right? And we could see how's it changing for other crew members that go up into the ISS.
Okay, so what findings from that mapping should mission medical planners view as an urgent public safety alert?
I think the genes that we found that seem to confer some resistance would be ones that we could keep an eye on. But there's nothing urgent, because no one got sick from that species. No one has been developing a disease. but it shows that the spaceflight environment does create a unique selective environment for how these organisms were evolved, and it can help us when we look at terrestrial species, how they evolve. So we can look at the same genes, look at some of the same pathways, and how they adapt. But there's nothing urgent for public health quite yet. But we do want to keep an eye on a lot of these changes.
Okay. Um, a very recent paper from your group looks at using microbes for upcycling plastic waste on the International Space Station. This flips the script from microbes as a threat to microbes as a solution. Can you tell us about this project and the vision of using engineered biology to create self-sustaining life support system?
Yes, yeah, this is a dream for the lab. Is a lot of the work we do is on mapping and characterizing and understanding, but eventually the goal is to do that for about 10 to 15 more years, but spend the latter part of our lab's work in my life, for
Hmm.
that matter, on using the catalog of genetic tools that we've mapped and curated to put them to work, right? So for in this case, using a species that's been slightly modified to absorb, digest and basically process waste, and upcycling, which I have a couple of grad students also working on other variations of this idea.
Hmm.
But we worked at this with Ariel Ekblow and also with some of the team at MIT that worked on this. So we wanted to, because we were a part of a larger team working on this together, but what made it such that we could, to a large degree, help process and digest the plastic and reformulate some of the hydrocarbons and molecules to be more useful. So it was a proof of principle. So it doesn't mean
Hmm.
that we have, completely absorbed all the waste and we're ready to go to space, go to Mars, but it shows that it was possible and that it's possible to do it in orbit. So that was the other thing. It's not just an in vitro study that's here on Earth, but also made it very possible to do it there, on the space station. So that we're hoping to do a lot more of that for these upcoming missions as well.
Okay, so ultimately, how do you see the astronauts' microbiome being integrated into the design of future bioregenerative life support systems, Bliss? Will future spacecraft architects need to design chips that don't just support human life, but also actively cultivate a healthy microbial environment so that a human becomes part of the system, not a beneficiary of it?
Yes, absolutely. So I mean, one of my favorite recent descriptions of this is that life doesn't happen on a planet. We think about where we'll find life again in the universe. It's often thought that life happens, or I think a better way to say it is that life happens to a planet. Is that, for example, life modified our atmosphere to create all this oxygen, right? So which dramatically changed the ecosystem and led to massive changes. So I think the idea of life happening to an environment is a more recent framing I've been using, because when you move into a house, you change the microbiome. When
Hmm.
you move into a space station, everywhere you go, subways, for example, we've had a large scale study looking at subway microbiomes for 10 years. They all move and change and are dynamically integrated. So we now that we can see it, we have to think about how do we engineer it. So
Hmm.
before we couldn't see it, it was too expensive, it was too difficult. Culturing was maybe the only option. But now we have large scale, high throughput means by which to map, monitor, and potentially even modulate what's present in our environment. So we still probably have some discovery work to do, but we've been even doing this. There's a study we're doing with Yale right now. Is if you flow air through a green wall of
Hmm.
plants, for example, does that make the built environment microbiome safer? Do people get sick less often? So we're doing a study to see how does the change of the environment help the health of the people that live and work in that environment, which is still ongoing, but is interesting, to start to look at these questions. So we still have some basic studies to be done, but we know, conceptually, these are real dynamics and can probably change toxins in the environment, processing processing of the atmosphere at the very minimum, because obviously it's
Hmm.
making oxygen, but you want to think about how you make that oxygen and what density and what speed. So yeah, that's very much what we can and should do for these future missions.
Okay, so your work is contributing to massive data repositories like the Space Omics and the Medical Atlas or SOMA. You're essentially building something like the Google Maps for human biology in space. How does these data banks from SOMA to your ChemBank at Cornell help you create a biomedical roadmap that can predict risks for future astronauts?
Yeah, we want this to be a lot like the UK Biobank is for predicting metrics for risk for astronauts. And so where we're at is in the very early stage. If you remember when the first human genome got finished in 2001, then it got again finished in 2003. It took a while. It really took 20 years to go from having one genome to doing millions of genomes, or even from going from one genome to start to do genome-wide association studies. That took really 7 or 8 years. So we're in the very early days because we, are just starting to collect some of these astronauts and across different missions. but it's a collaboration and coordination with groups like NASA, ESA, JAXA, commercial spaceflight flyers. So it's a really it is a early stage, but we want to make it so we can have the data be longitudinally collected, annotated, and contribute towards this edifice of knowledge that makes it so you can predict: are you at a high risk for spaceflight sickness? Are you at a high risk for, do you need different nutrition, different medications? The precision medicine for astronauts is in the very early stages, but this is the bedrock of it, is what we're trying to build out. And then we want to not do it alone, though. We want to do this in partnership with as many people as we possibly can, because it is a large scale problem that will require a lot of large scale coordination for sure.
Okay, so these data banks have a notable population gap. There is no data for individuals under 18. The youngest to go to space were Oliver Damon and Anastasia Myers. They flew on Blue Origin suborbital missions and were 18 years old when they did that. Now, as we seriously contemplate multi generation missions or at least a permanent settlement, how does this lack of data on developing biology in space influence our ability to plan for families or young people living off Earth?
Yeah, that is a good point. We have zero real data on there, so it means it's a big gap. It means we have some time, because I don't think we'll start to see families of four with their kids going on vacation in space, in the next year or two or even 5 years.
Mhm.
But 10 or 15, 20 years from now, if it really gets routine and safe and inexpensive to go to space, we will see people who want to bring their kids and they want to go to space, and by all rights they should. So that is something we have to expand out where we're looking in the databases, and so that is one of the big challenges for sure, that we have to build out that data. And right now there's no it hasn't happened yet. So I think there's different rules, of course, for children. You have to get an assent form, you have to get other consent from the parents, but but this is something that can and should be done, because we'll need to know if we start to have this spacefaring civilization where we go back and forth between the moon and the Earth and hang out in space for sure.
So have you contemplated, like, imagine putting a teenager, like, you go to up to space and your hormones and your body changes. If you put a teenager with all those hormonal changes they're going through up in space, uh, it will wreak havoc. We can't even imagine what could happen. Do you have like a theory or hypothesis how things could be, or this is still really unknown?
Um, we really don't. I'm trying to think. We just don't know. There's even some people who want to look at taking early embryos into space, or looking at pregnancy in space. That's
Hmm.
even more unknown. But if someone say 16 or 17, they'd be very close to an adult in a lot of ways. In many ways not, but they still, it'd be much different from someone who's like prepubescent, like 11 or 12, or just in the middle of puberty. Would, I think, be a much more volatile time. So I would personally feel probably more comfortable starting with like 16, 17 year olds first,
Hmm.
maybe start to look there. But then once we get to puberty or prepubescent children, everything's unknown, because we just have no idea what to expect. So you'd probably do something similar. We start with maybe a suborbital flights and start
Hmm.
very simple. Start with, maybe some parabolic flights. Although that's not that much different from Earth, but start small and start safe and start, closer to Earth, I guess it is. So if it is orbital, it's only a couple days in orbit, let's say, for example. So I think, all the ethics around that, like how do you fully consent a child to something with a vastly unknown risk? Like we don't know, but if someone wants to buy a rocket and go to space, they could do so. So we need to also, if someone's going to do it, we need to help them do it safely. So there's a tension there, but one that I think is going to get teased out eventually, and eventually kids will fly if we do this right.
Great. Let's ground this question. How does this gap reshape the ethical and scientific research questions we need to prioritize now?
I think it shows that one is just accessibility to data. We've tried to really consent crews to open their data out for people to use and to mine, and that is ongoing. And we want it so people can really have you can build models, right? I think one example is, for example, risk for diseases. We're still understanding which genes contribute to disease, which are very clearly defined, or even height, for example. One of the most heritable traits that humans have, there are hundreds of genes that contribute. So it's a complicated multigenic phenotype or multigenic trait that we
Hmm.
have to figure out. And so some of it is just giving people data to build better models, which will take time. For diseases that are well established, or traits like height that we know very well, we're still learning. So it's going to take. I think the other thing is people need to know that this will take several, probably years or even decades, and that it will only make progress if people, I think, bravely share their data with others to build on. So you don't have to, of course,
Mm-hmm.
with any data set. If you consent to a protocol, you can share all of it or none of it, or it's up to you. But
Blind. Yeah.
I think that it's nice when people share, because then we can use that data, of course. But you have to be secure and safe and consented, of course.
Okay, so now let's move on and let's talk to countermeasures. The nutritional tweaks, drugs, and microbiome engineering. If you had to pick the top two interventions to deploy on a 2-year Martian surface stay, what would they be, and what's the biggest unknown for each?
I think we've done microRNA countermeasures. We're looking at small molecules like Chemferol, for example. We're working on that with a couple of companies that have synthesized products that we want to try. And
Mhm.
a lot of this is starting, of course, with in vitro, so just doing this in cell culture or in simulated environments. But we've flown some of them in space and are beginning some of those kind of experiments. So it's early, but I think we have to do three broad areas of countermeasures. So one of them is pharmacological, which people are pretty familiar with. You take a drug, it helps protect you. That's great. So it's a specific drug. The other ones are biological. So you take like a probiotic. You take maybe it's a small peptide that's just a natural product that you could use. So it could be something that's biological versus pharmacological. There's also along with biologicals, also just like the physical, your barrier of your skin, the barrier of the spacecraft. There's really mechanical protection. I guess would be almost like maybe a fourth category. But the final category would be, I think, genetics. So you can think about ways to activate DNA repair genes before you need them. So if you know a high radiation burst is coming, you're prepared for it. So this is the more kind of the thesis of my first book, which is that we should consider all these options when looking at sending humans to faraway planets. The argument is that if you send someone to a planet and you know they're going to be at an extremely high risk of radiation, and you have a means to protect them genetically and physically and pharmacologically, and you don't, then that would be unethical. But it assumes that it's safe, efficacious, and well established to use genetic tools for protection. But it's very early days, except that now that we've seen FDA approved trials, we have it working in our laboratory. So I think what used to be very hypothetical 5 years ago is now actively deployed.
Okay, so this leads me to a more science-based, futuristic side of your work, engineering life for space. You've demonstrated that inserting a gene from a tardigrade, the DSUB gene, can make human cells 80% more resistant to radiation, in theory. You're also exploring enhancing the p53 tumor suppressor gene. Um, this is a move from protecting the astronaut to upgrading the human. How do you decide which parts of the human genome are bugs to be fixed versus features to be enhanced? And maybe quickly, can you talk about the DSUB gene and the P53 tumor?
Yes, the DSEP is the DNA damage suppressor gene that was identified in tardigrades, or these water
Hmm.
bears that can survive in the vacuum of space. And what's extraordinary is they have shown that they can be rehydrated. They are very robust creatures. And when you take that protein, there was a transient transfection that was done by Hashimoto back in 2016 that showed it was possible. So we've made a stable line that expresses this protein and also gives this reduction. So it shows that you can take a protein made from a different species that has an evolutionary lesson and transfer that lesson to a human cell. And so that's where I think we could transfer the protein, or we could transfer the gene itself to make it stably expressed, and use those tools to help us survive. So I think all creatures are fair game for in terms of giving us ideas and
Hmm.
physical molecules to help us stay safe in high radiation environments. And then also, for P53, that is the one of the genes called the guardian of the genome, because it really helps with DNA repair and protecting our DNA. And we've been thinking about ways to modify and have started modifying that gene with targeted therapies, just targeted gene therapy. So they're doing. It's very early though. The question of which of them are bugs or which of them are features depends on the planet you're in or the environment you're in. So I think we don't. If it's really, they may help you with DNA repair, but what if they make it so that you live 10 years less, something. So a lot of the trade-offs we don't know yet. We say, okay, this protein seems to be helping with DNA repair and DNA damage, great, but are there other caveats and genes that change? We've published and we know that there are other changes, we just don't know their impact yet. So it's too early to say what's a bug or a feature because of pleiotropy, because one gene can have many functions and we don't know yet. But that's why a lot of we do in vitro studies first. We start in the cell, we see how things function and try and tease it out. But then eventually we will, need to try to monitor this in missions that are in humans for a long period of time.
Okay, so CRISPR programming and protective genes, like we were talking about, the DSUP and P53 are tantalizing. In your book, you make the argument that engineering ourselves to live in space is a human survival eventuality, a no-brainer for a long-term planning for the survival of the entirety of the human race, especially with the sun engulfing Earth in a couple of billion years. What ethical or safety guardrails do you see as a non-negotiable before we edit human cells or microbes for space resilience and adaptation?
I think all ethical questions are clear through the lens of a billion years. So if we debate ourselves into paralysis and we don't do something, then we'll just burn to a crisp on this planet, and probably in about 500 or 600 million years, but for sure by about a billion years. So I have this real clarifying view towards what comes in the long run, and that life, as far as we know, is rare in the universe, or as far as we know, only on this planet, and to preserve it and to preserve any of our values that we care, including the value to question and to come up with new ethical frameworks or science experiments or poetry or music or anything that we like and value. We have to at some point begin to go to other planets. And so if we don't need any of these genetic tools and we somehow are more resilient than we thought, and we can survive in lava tubes on Mars, and we can make our way with artificial gravity and electromagnetic fields that keep us safe, great. But
Mhm.
if we have not succeeded, and we have genetic means to do so, I think we should deploy them. So the non-negotiable is if it's taking too long, and we think we have an efficacious means by which to keep humans alive, either through so some of the ability, for example, to make all of our own amino acids or to make vitamin C in our genome. We know we can do that because we've done it before, and other primates have done it. But we could, that would make it so we could survive in these long missions. And so if we find ourselves being too gun shy with some of these genetic tools because we're worried about what would happen long term, but then we end up dying on the planet, it's all moot, right? So
Mhm.
if we are stuck here and the debate paralysis left us here, then we don't have the value to even debate anymore. So I find it to be that it's kind of an existential question, that the existence precedes the essence of anything, as Jean-Paul Sartre would say.
Yeah, I think a lot of people, first thing when they think about genetic engineering the human body, they think the Wrath of Khan for Star Trek.
Yeah, exactly. Yeah.
Well, yeah, okay. So beyond editing human cells, you've also discussed engineering microbes to live in our, let's say, on our skin to act as a biological sunscreen against radiation. Is this a more ethically palatable and technically feasible near-term approach to biological enhancement?
Yeah, it's also much more transient and portable. You could take it and move it, or you could remove it, or you could modify it more easily. So they're both ethical if they're needed for survival. And so if something is safe and efficacious, you should use it. If it's not, you shouldn't. But I think if something can work to help keep you safe or healthy, then you should use it. But that is another mechanism we're looking at, because it's very portable. It's
Mhm.
transferable. You could take it on, take it off. You could modify it. So that's still earlier stage, but that's definitely something we're working on.
Awesome. You're also at the forefront of using emerging technologies like single cell omics, AI, and organoids. Of these powerful tools, which one do you believe holds the most promise for accelerating the discovery of new countermeasures, and why?
All the above, I guess, is what I want to see, because and the AI has even been designing and critiquing experiments. So probably AI, and I even think organoids will change a lot in the next 3 or 4 years, because you can have many kinds of organoids that can do a lot of different types of modeling for human systems. So I think even the what we'll see for the types of organoids will change, and how you use them. So I think that's still early-ish, but I guess both. Yeah. So whenever we're given a couple options, I was like, well, both. But the AI would probably be the most driving feature, because we can mine so much data and process it so much faster than we could before, which is pretty powerful.
Okay, let me ask you a question. I've heard that, like, last time I was talking to Professor Afshin Beheshti, he mentioned that the mitochondria used to be a separate organism. Well, there's a hypothesis that the mitochondria used to be a separate organism, and then it joined with the cell with
Mhm.
our cells to become an organelle. Do you think this is also could be a possibility where we can join other organs or organelles into to help us improve our living in space?
Yeah, actually, I even have an organelle wish list. Chloroplast, for example,
Hmm.
would be great to include in human cells. There's a section in my first book I described the bioenergetics of that.
Hmm.
That would be great. I would love to see organelles that have functions for metabolism or DNA repair that are more specific to these functions. So, yeah, the mitochondrial collaboration with eukaryotic cells is the longest collaboration that human cells have. It's been billions of years, so it is a great coordination. But still, I think what's also fascinating is a lot of the genes that were originally in the mitochondria have now migrated and are part of the human genome and
Hmm.
get copied by the human genome. There's a small subset, just handfuls of genes left that are in the mitochondria for its replication, but they're still there. So, at some point, seeing this exchange of mitochondrial genes that have been now become part of humans, it's really a longstanding collaboration, but it's still evolving. Actually, it's still changing and evolving.
Cool. So the biological challenges of a short lunar mission seems vastly different from a multi-year Mars mission.
Hmm.
What is the single biggest biological problem we need to solve to get to Mars that isn't as critical for a return to the moon?
I think the one most people are the most concerned about is radiation, but there's also a lot of concerns about cognitive capabilities when you're in such a small capsule for 6 to 7, 8 months. That is a big concern. The psychosocial, the stress on the body with that amount of microgravity for that long is also. We did it with the Kelly study with the twin study, but you'd have a lot more radiation in that environment, so it might be slightly different. So those are some of the biggest factors. But radiation is probably the one we think about the most. I think getting there would be the hardest part. Once you land on Mars, you could go underground, and it'd probably be less of an issue. But it's the interplanetary space that we know is going to be a bit rough.
Okay, speaking about landing on Mars, the astronauts' fast recovery in order to perform critical operations after landing is vital for any planetary mission. And I'm talking about recovery in minutes, not days. Um, we were lucky that the moon we are lucky that the moon has such a low gravity. Our astronauts were able to do a moonwalk soon after landing. On the other hand, our astronauts that have spent time on the International Space Station are definitely not able to perform critical operations directly after landing. What about Mars? How do we solve this? Is this a technology problem or a biomedical problem?
It could be both. I mean, so to your point, though, if you spend 6 months in microgravity and then you land at 3/8 gravity, you should probably be able to function, but it will still be hard on the body. So you're suddenly going to have a decent amount of gravity, but it will be easier than being on Earth for sure. So, but then you're also in a very stressful environment that has almost no atmosphere. It's on literally another planet where no one can help you. So there will be other stressors there as well. But I think I think that will hopefully should be manageable because of the 3/8 gravity. When they land, they can hopefully perform tasks okay, we think.
Okay. Chris, you wrote The Next 500 Years, the book, in 2021. ChatGPT was released in 2022. Exponential advancement across the board that it made. It is now mid 2025. If you were to issue a new edition for your book in 2026, what one or two things would you update? Would you update timelines, milestones? Would you add new technologies or remove outdated science research?
Hmm. I think it would add a lot of the newer technologies. Some of them, for example, in the book, I talked about prime editing, and I wrote in the book that there will invariably be other gene editing systems that are discovered, and indeed they've come to pass. So, like the omega proteins from Fengzheng is already found and published. And there's other predicted new gene modification systems, actually dozens of them that are possible. So we think there are many other ways to create modified genetic systems. So I talk about that. I would also add a bit of some of the synthesis tools that have come along of how you could synthesize whole genomes, different new technologies on enzymatic synthesis that have come out. But also, I would update at the time we didn't have the commercial space flight sector was just getting started. So I think now it's going to probably be if things go the way things are planned, the commercial space flight sector will probably eclipse most of the data and samples that NASA's made over the past 50 years. It'll be eclipsed within 4 or 5 years. So it'll be a rapid uptick of the number of space stations, spacecraft, and missions possible. So I'd probably update some of those timelines too.
Okay. So You've coined the term planetary liberty, the idea that engineering a human to be perfectly adapted for Mars might make them unsuited for Earth. How do we balance the moral duty you've spoken of to preserve and propagate life with the ethical duty to protect an individual's freedom to live wherever they choose?
Yeah, the advantage of this is you can do epigenetic editing, where you turn genes on and off, like we talked
Hmm.
about a little bit earlier, that lets you adapt what your genetic profile looks like. Now, again, this assumes that you need to do that. You might not need to at all. Maybe the body adapts and you're just uncomfortable for a few days.
Hmm.
But if you really are marooned and perfectly, created to survive on Mars and are unable to visit Earth, I would consider that a failure. That we had not maintained the liberty of people to be able to choose where they'd like to go. That's the whole idea of spacecraft and exploration, is that of course, you have to modify the container in which you travel, but you want to travel. You want to be able to go and explore and live where you'd like to go. And so, yeah, the planetary liberty is the idea that you could use epigenome modifications to tweak your gene profile so you could survive in the mountains or a different planet wherever you'd like to go. Which again is very hypothetical. We've not done that yet, and again,
Hmm.
it may not be needed. But as a concept, if the kind of planetary liberty is something that I really want to hammer home, is that is the ability to transit the solar system, someday the universe, is powerful, and is something that we take for granted on Earth, that you can just get on a boat, get on a plane, go anywhere you'd like and see. And that is a very important freedom, though, that we should preserve, not just on this planet, but when we go between planets.
Okay. Let me ask you this. Wouldn't creating those kind of genetic and epigenetic modification, or do you consider that this could create like planetary geopolitical chasm entrenched in identifying as a Martian versus a human? Like I'm talking like the Belters from the book and the TV series The Expanse comes to mind.
Yes, yeah, it's a great book, also a great series, and I think so. What's interesting about that book and the series is, I it's an extraordinary vision of the future, where we have people routinely and easy and seamlessly moving between the planets of our solar system with advanced propulsion, and even though we have that extraordinary technological leap, the nations of, or the basically the Mars and terrestrial and Belters uh, factions basically are just as petty and backstabbing and competitive as they are, like, with countries here on Earth. So
Hmm.
I was sad when I first read that book, because I thought, no, if we have such an amazing future, wouldn't people work together? But maybe that is a more Machiavellian, maybe more accurate view of what would happen, is that people would just start to be competing in that regard. So I hope not,
Hmm.
but a reasonable reading of history would indicate that that's probably what would happen on some level. Is that resources are limited, people are competitive, people are functioning either based on fear or love, if you go back to Machiavelli, and that this has always happened with humans. So either we'd have to dramatically change how society works, or humans may have to change, but there's no evidence that would be, sadly, different from what was described in those books. But I would endeavor to make it different. I'd say, like, that we'd use means to have people work together and enable people to move easily between the planets.
Okay, Chris, everything we just talked about, it sounds like a science fiction, but it has very real applications on Earth. Your company, Cosmica Biosciences, is built on the insight that spaceflight mimics accelerated aging. How is studying astronauts helping you develop therapeutics for age related diseases like Alzheimer's or cardiovascular diseases right here on Earth?
Yes. So this is the idea that we see a lot of, like, markers for osteoporosis, muscle wasting, some of the other biological markers of aging. We see change in flight. So we're using spaceflight as that accelerated model. Much of the what's in the company is still private, so it's not out yet. But we are exactly using these kinds of models to have what could be different targets or different small molecules that could affect how the body functions or how it's aging. But looking everywhere, from mitochondria to muscles to bone to brain health, because we can study a lot of those things pretty easily right now. So I think we have not yet launched the first product, but things are coming along, and yes, stay tuned. I guess I'd say it's still early, but it's stay tuned.
Cool. Your lab applied genetic forensics to archaeological artifacts on Earth. Tell us a bit about that, and how did the protocols optimized for the International Space Environment unlock new stories in ancient DNA?
Yeah, in ancient DNA, we worked with Oded Rachavi to look at the Dead Sea Scrolls, for example,
Hmm.
which we wanted to use genetics to reconstruct some of the pieces of scrolls that hadn't yet been linked together. by using sort of genetic guided artifact reconstruction. And in that case, we were able to actually show that from some of the caves where these Dead Sea Scrolls were connected, that we changed or proposed changes to how they should be stitched together, and also we're able to confirm the animal hides that were used for some of the scrolls that were that each would come from a different animal. So we could actually use the genetics of a piece of one animal that was part of the substrate for writing to confirm that that piece was from one animal, which means it's more likely to be together. So it's a fascinating utility of genomics for artifact reconstruction from something that's even biblical import, which is a fun project.
Okay, so let's talk about your work in metagenomics for planetary health. The same deep sequencing tools designed for the astronauts now map pathogens in urban sewage or hospital wards. How would Metasub changes terrestrial public health?
We've been using Metasub, which is the metagenomics and design of subways and urban biomes, which is called Metasub because it's shorter. But that is a large-scale international consortium that's been out swabbing and monitoring urban environments plus wastewater and hospitals for more than 10 years now. And we use this as a way to look at the movement of antibiotic resistance, look at new viruses, look at some of the changes in the ecosystem. And during COVID, of course, we used it to track during the pandemic where the virus was going, what strains of the virus were popping up. So it was a very large scale effort that's been ongoing. It gives us kind of a Google Maps for microbes in the cities. You can see what organisms in what place of the world at what time. And so it's not yet real time, but it does, it gives us a really good map of what's in the world around us, which has been a lot of fun. Yeah, and also as a good outreach. Like we get students that go out and swab once a year, and they learn how to go out in public and swab and learn about metagenomics. So it's both an outreach program as well as a science program, I'd say.
Very cool. Another thing your lab is utilizing is conservation genomics. In this context, can you share with us about your work with the black-footed ferret and how this work fits into the greater scheme to one day conserve humanity on an Earth-like planet in a different solar system?
Yes. We've been working with Revive and Restore and some of the Colossal Foundation to sequence the black-footed ferret, which was recently cloned, and to help get a better reference genome and help preserve the genetic diversity that's known for that species. So we do both genetic and epigenetic profiles, so looking at the changes of those creatures, and we mostly do it just as a courtesy because we want to help with conservation efforts. We had just recently got some support from the Colossal Foundation for other expeditions to do some of these collections. So we want to very explicitly help in that regard. I feel like every genome that's lost to history is one that we have to work hard to resurrect and de-extinct later. But if we could, keep it alive now, that helps us for understanding the diversity, genetic diversity, conservation efforts. So, it's an ongoing project, but we have to get that paper out shortly. But it is we have a lot of the data and can see that the cloning process actually works pretty well. So we want to support that for future species as well.
Great. Looking forward to it. Chris, before we wrap up, is there anything else you'd like to share with our audience or advice you'd like to share?
No, I think that's we that covers a lot of what's in the lab. There's some other projects, of course, ongoing, but and some of the clinical. I guess the thing I'd anchor on is that a lot of it seems exploratory or in looking in space and these things that seem abstract, but they a lot of the exact same methods we do for cancer patients. We do it for rapid diagnostics of infectious diseases at the hospital. So these methods, while they might seem like they're abstract in space, are really already being utilized on Earth and also can help advance a lot of terrestrial medicine. And my wife was making fun of me. She's like, "I heard you on the phone saying terrestrial medicine, but what does that mean?" I was like, "Well, it's just anything on Earth." But she's like, "Isn't all the medicine on Earth?" I was like, "Well, most of it, but not all of it." So this idea of having a phrase called terrestrial medicine is extraordinary. The fact that we can say that and that actually makes sense. And so it's a really unique time. But I want people to think about how they can get involved and start to jump into the research and help out because we are, really becoming a spacefaring species. So it's excited to plan for it and have people get involved.
Chris, it's been an honor to have you here. Thank you so much for sharing your insights and expertise.
My pleasure. Thanks so much.
To everyone else listening, this is the Omar Alayli Podcast. Thanks for joining us and keep exploring.